AS Level Biology 9700, Paper 1, Variant 2, February March 2026. And in this video, we are going to discuss from question number 1 to 13. So, question number 1 is on magnification. The eyepiece reticule for a light microscope at 100 magnification was calibrated using a stage micrometer scale as shown in the diagram. Each small division on the stage micrometer scale measured exactly 0.01 millimeter. So stage micrometer scale, eyepiece reticule, you can see them over here. So stage micrometer units are, you can see 1, 2, 3, 4, 5, they are equal to 34 and they are equal to 100. So according to this, I would like to say that 100 eyepiece reticule units are equal to 34, or then we will multiply this 34 because it is equal to 0.01 millimeter. So we will multiply it to 0.1 so it will become 0.34. So I would like to say that 100 eyepiece reticule units are equal to 0.34 stage micrometer scale. So then we will divide this 0.34 with 100 to calibrate them. So it will become 0.0034 millimeter. So it is up to you. You can convert this millimeter into micrometer now or after completing the answer. Now you can see the length of the measured, it is 47 units. So we will multiply 47 with 0.0034. So it will become equal to 0.1598, and when it will be multiplied with 1000, so it will become, after multiplying with 1000, the answer will become 0.1598 multiplied by 1000. So it will become 159.8, which is very close to the, it can be 1.6 into 10 raised to power 2 micrometer. So it is very close to this answer. So C is the correct answer. Now we will move towards the question number 2. And question number 2, one of the electron micrograph shows a eukaryotic cell. The other shows a prokaryotic cell. So here you can see what is the approximate distance across the eukaryotic cell at its widest point. So in this case, you can see in this question number 2, here you can see we have to choose which one is the prokaryotic cell and which one is the eukaryotic cell. Here you can see the cell on the right side is, this one is eukaryotic. And this one is the prokaryotic. Because it contains a distinct membrane-bound nucleus and visible internal organelles. You can see it has a membrane-bound nucleus. So its magnification is given, which is 4000. So what is the approximate distance across the eukaryotic cell at its widest point? So widest point if we measure the slightest point, uh, you can do it by yourself on your uh, paper, which you can choose. According to me, it is nearly equal to 84. So when we calculate the answer, then it will be, the magnification is equal to m length over actual length. And if we want to find this, so image is 84 divided by 4,000. So the answer could be 4,000. It is equal to 0.021. Now we have to change it into micrometer. So we will multiply it with 1000. So it will become equal to 21 micrometer. So the answer is D. Now we are moving towards the question number 3, and question number 3 is, which polymer is a major component of the cell walls of the typical prokaryotes? And they have given us 4 options: cellulose, peptidoglycan, and glycogen, and suberin. So cellulose, it is found in the plant's cell walls. So this is not the correct option. It is not found in the prokaryotes. So you know that glycogen is a storage carbohydrate in animals and fungi. Again, this is not the correct option. And suberin is a waterproofing polymer which is found in certain plant tissues like the Casparian strips, So this is also not correct option. So only correct option which is left behind is the peptidoglycan. So the typical prokaryotes have cell walls which are made up of peptidoglycan, which is also known as murine. So B is the correct option. Now question number 4 is there, and question number 4 is, which features are shown by prokaryotes cells and also by eukaryotic cells? And students, you know that question number 4, the correct option, cell membrane, first option is the cell surface membrane, which is present in all cell types. So whether they are prokaryotes or eukaryotes, so the cell surface membrane is always there. Lysosomes, they are the single membrane bound organelles found only in eukaryotic cells because they contain the hydrolytic enzyme. So lysosomes are not found in both of these type cells, only present in eukaryotes. Cytoplasm, again, it is present in all cell types, that is whether they are prokaryotes or eukaryotes. And then 70S ribosomes contains only, the prokaryote, it contains 70S ribosomes. So eukaryotes have 80S ribosomes in their cytoplasm. They also contain the 70S ribosomes inside their mitochondria and chloroplast. Therefore, this feature is shown in both. So the 70S ribosomes are present in both. So according to this, the 1, 3, and 4, they are the correct uh, option. So according to this, B is the correct option. A, C, and D are not the correct options. Now I would like to move towards the question number 5. And question number 5 is, some proteins synthesized in plant cells are modified before they are secreted. Which cell structure in plants is responsible for modifying proteins before they are secreted? You know that students, you know that uh, plasmodesmata. First is the plasmodesmata. They are the, you know, the channels. They are microscopic channels uh, between two cells. You can say through the plant cell walls. So they are not helpful in modifying the proteins before their secretion. So tonoplast is the membrane which surrounds the vacuole, central vacuole in plants. Then Golgi bodies. Golgi bodies is responsible for this modifying proteins. So adding protein chains to form glycoproteins and packing them into vesicles or secretions. So Golgi bodies are the those organelles which are in plant cells which modify the proteins before their secretion. And the left behind lysosomes again, they contain hydrolytic enzymes for breakdown, not secretion or modification. So C is the correct option. Now question number 6 is there. Question number 6, which row shows the types of the membrane that surrounds the mitochondria and the nuclei. Student, obviously the answer will go the B, because you mitochondria is double membrane structure, outer and inner membrane. So nuclei also they have the double membrane. Nuclei are closed in nuclear envelope, which is also a double membrane. So I would like to say that B is the correct option and all other options are not correct. Now question number 7 is there. And question number 7 is, it is suggested that primitive prokaryotic cells may be ancestors of certain organelles in eukaryotic cells. Which organelle is most similar to a typical prokaryote? Golgi body, lysosome, mitochondrion, and nucleolus. So, I would like to explain according to the endosymbiotic theory, which you have read in your book. So, you know, this question is about this theory, which suggests that semi-autonomous organelles like mitochondria and chloroplast evolved from primitive free-living prokaryotic cells like typical prokaryotes. Mitochondria contain their own circular DNA loops, have 70 S ribosomes, and reproduce via a process similar to binary fission. So, according to this endosymbiotic theory, the answer is C. Golgi bodies are used for modification. Lysosomes are hydrolytic, contain hydro, and nucleolus is present in the which has a different function instead of this one. Now, question number 8 is there. Which of the sugars, fructose, glucose, and maltose are reducing sugar? So, you know that the monosaccharides, including both glucose and fructose, are reducing sugar. They have a free uh, aldehyde or ketone group capable of producing Benedict solution. So maltose is a disaccharide made up of two glucose units linked by an alpha 1,4 glycosidic bond, leaving one of the reducing ends free. So it is also a reducing sugar. Sucrose is a common example of non-reducing sugar. So according to these, fructose, glucose, and maltose all are the reducing sugar. So according to this, C is the correct option. Now question number 9 is, which molecule in a muscle cell is rapidly hydrolyzed to produce glucose? Definitely amylopectin, amylose, they are not the, they are not producing the glucose. Cellulose is also a carbohydrate, which is, you can say, the make up the wall in plant cells. And only the glycogen is the primary storage polysaccharide found in animal cells, heavily concentrated in liver and skeletal muscle tissues. So it is highly branched, even more than amylopectin, containing numerous terminal ends that enzyme can rapidly target for hydrolysis to release glucose monomers whenever energy is needed during muscle contraction. So amylose make up plant starch while cellulose forms plant cell wall. So amylopectin, amylose and cellulose are not the correct option. Only D is the correct option because these are the part of the animal cell, whereas amylopectin, amylose, cellulose, they are part of the plant cell. So glycogen is a storage material in the animals, or a muscle cell is the cell, animal cell. Now question number 10 is there. Question number 10 is, so here you can see that the question refers as, the diagram shows how the beta glucose monomers of cellulose are linked to each other. So according to this, they have given us a chain. The OH groups on carbon 2 of the adjacent glucose monomers are on opposite side of the cellulose molecule. So what is the main way in which this arrangement of OH groups on the carbon 2 contributes to the strength of the structure made of cellulose? So first of all, I would like to go on options A. The OH groups on both sides of cellulose molecule can form cross links within the same molecule through the formation of glycosidic bond between adjacent OH and CH2OH groups. This is incorrect because they state the cross links are formed by glycosidic bonds. Glycosidic bonds only link individual glucose units together within the same chain, not between different chains. So according to this, A is not a correct option. On each side of the cellulose molecule can form cross links with different cellulose molecules. Again, they are talking about the different cellulose molecules. So B is again not the correct option. Now I would like to go on C. The OH groups on both sides of a cellulose molecule can form bonds within the same cellulose molecule through the formation of hydrogen bonds between adjacent OH and CH2OH groups. So this is also incorrect. It suggests that the strength comes from hydrogen bonding between the same molecule, whereas the structural strength comes from linking different molecules together. So this is not the correct option. So the OH groups on each side of a cellulose molecule can form cross links with different cellulose molecules through the formation of hydro bonds between nearby OH group. So it is correct that the groups OH group form cross links with different cellulose molecules via hydrogen bonds. So this is the correct option. That is, it happens. Now question number 11 is there, and question number 11 is, which statement relates the structure of triglyceride molecule to their function. Triglyceride molecules can function as energy stores because they have a large number of CH bonds in their fatty acid bonds. So first of all, I would like to discuss the structure uh, of these triglycerides. So they are composed of glycerol molecules joined to three fatty acid tails via ester bonds, you know well. Now the fatty acid tails contain long hydrocarbon chains with a very high proportion of carbon-hydrogen bonds. So when triglycerides are oxidized during respiration, the breaking of these bonds releases a large amount of energy per unit mass compared to carbohydrates. So this makes them highly efficient long-term energy stores. So according to this, the A is the correct option. Now B, uh, molecules can function as energy stores because the large amount of energy are required to hydrolyze ester bonds. The energy stored is released during the oxidation of hydrocarbons, not from the hydrolysis of the ester bonds. So this is not correct. Now, C molecules can provide energy to all the cells of the body because they are non-polar molecules that easily dissolve in water for transport in the blood plasma. Triglycerides are entirely non-polar and hydrophobic, meaning they are insoluble in water and cannot really dissolve in blood plasma, or simply simply they can say triglycerides molecule can provide energy to all the cells of the body because they are polar molecules. They easily through the phospholipid bilayer. They are not dissolved, and they are not, you can say, simply diffuse through cell membranes without transport lipoproteins. So again, D is not the correct option. Only A is the correct answer or the option. Now move towards the question number 12. And question number 12 is membrane fluidity is affected by temperature and by the percentage of fluid fatty acids tails in the phospholipid molecules that are saturated. Which combination of these two factors would result in the greatest membrane fluidity? So they have given us two columns, one for temperature, the other percentage of fatty acids details in the phospholipid that are saturated. So first of all, I would like to go with the discuss the temperature. As temperature increases, the kinetic energy of the phospholipid molecules increases, you know. So they move faster and vibrate more, which increases gaps between them, resulting in greater membrane fluidity. So therefore, 40 degrees temperature promotes higher fluidity than 10. So only 40 degree can be considered as the correct temperature. Now, the other is the percentage. So first of all, you know, saturated fatty acids have straight tails that pack closely together, making the membrane more rigid, no double bonds, no triple bond. So unsaturated fatty acids have kinks so that prevent close packing, keeping the membrane fluid. So to get the greatest fluidity, you know, need a lower percentage of saturated fatty acids. Thus, 20% provides greater fluidity than 80%. So B is the correct option. Now, question number 13 is there, and question number 13 is the solubility in water of different regions of the cell surface membrane changes with the distance of each region from the center of the membrane. So which graph shows the approximate relationship between the distance of each region from the center of the cell surface membrane and the solubility in water. So according to the statement which they have given, I would like to say that only the graph A is the correct. B, C, and D are not correct. Let me explain. So you know that the core of the cell surface membrane is made up of non-polar hydrophobic fatty acids, tails, because they repel water, the solubility in water. At the exact center, that is 0, must be at its lowest. So it will be lowest over here. This eliminates graph B and D. So it is highest in B and it is D. You can see it is also highest over. It should be lowest. So according to this, A and C, should any one of them should be the correct. Now I will move towards the inner and outer surfaces of the membrane consist of polar hydrophilic phosphate heads. So these regions interact easily with the aqueous environment inside and outside the cell, meaning their solubility in water is high. So the hydrophobic core formed a distinct continuous interior layer, while the hydrophilic heads form distinct outer boundaries. So graph A correctly models their structure with a sharp step-like increase in solubility, where the phosphate heads begin rather than a gradual linear slope. This linear slope, so this is not correct. So gradual linear slope from the absolute center. So you can see solubility decreases and then so this is the correct option according to the arrangement of this. As you know that the hydrophobic fatty acid tails are in the center, least soluble in water, and the hydrophilic phosphate heads are on the outer and inner surface of the membrane, more soluble in water. So according to this, all these, the A is the correct option. Now students, please like, share, and subscribe my channel and to watch more videos and to get notifications for other videos of the past papers. And thanks for watching.